Hello, my name is Rainer Bischoff and I'm heading the Analytical Biochemistry group at the Department of Pharmacy. I'm also responsible for the course Pharmaceutical Analysis C, which you are going to follow. If you would like to know more about the activities in our group, please consult the website given on this slide. The Pharmaceutical Analysis Course C builds on knowledge that you acquired during Pharmaceutical Analysis A and B, as well as during the Biostatistics course. The teaching goals of this course are to understand the role of bioanalysis in drug research and development and in biomedical research, to gain an insight into the most important instrumental bioanalytical methods, which will be introduced in the beginning by the first 6 lectures, to be able to develop and apply bioanalytical methods in different areas related to clinical chemistry, hospital pharmacy, and regulated drug analysis or the analysis of biopharmaceuticals. These parts will be given towards the end of the lecture series as applications. And finally, to be aware of regulatory requirements concerning method validation and quality as needed for regulatory development in industry and for bringing drugs to the market. Specific teaching goals comprise the understanding of the relationship between the physico-chemical properties of analytes and the methods that you need to analyze them. It is also important to acquire knowledge about separation methods such as electrophoresis and chromatography, of which you have heard during previous pharmaceutical analysis courses, as well as mass spectrometry, and very important method to detect and identify molecule. We will also talk about so-called ligand binding assays. These are assays to measure molecular interactions such as immunoassays and receptor binding assays. Finally, it is important to gain knowledge about the analysis of proteins, notably biopharmaceutical proteins, the fastest expanding group of novel pharmaceuticals to date. The lectures in part 1 will be given by me and are about the technology as shown here. You will be introduced to the concepts of bioanalysis, the important part of pre-analytics and sample preparation, because it is obvious that low concentrations of drug molecules cannot be analyzed directly, for example, in blood. You will hear about ligand binding assays, notably receptor and immunoassay, such as ELISAs. You will hear about the combination of liquid chromatography with mass spectrometry, so there will be a part on mass spectrometry and its concepts, and on liquid chromatography, which is a slight rehearsal of the Pharmaceutical Analysis B course, but very much applied to bioanalysis. We'll introduce you to the separation of macromolecules, which is different from small molecules that you may be mainly used to, like proteins. And finally, culminating in the analysis of biopharmaceuticals, bringing forward a few cases from real life that I have worked on and some of my colleagues have worked on. The second part of the lectures will be given by experts in various fields. You will first hear about quantitative bioanalysis in a regulated industrial environment. This will be given by Honorary Professor van der Merwel from PRA Health Science and the University of Groningen. You will be introduced to the applied pharmacokinetics and therapeutic drug monitoring, as well as toxicology, important application areas of instrumental bioanalysis. This will be given by Professor Tau from the University Medical Center in Groningen. and finally to clinical chemistry and the interpretation of laboratory data, very much related to pathophysiology, the diagnosis of disease, and the monitoring of response to therapy. This will be given by Dr. Dijkbrouwer from the University Medical Center. During this course we will use different teaching tools. First of all, the lectures, which provide you with a theoretical background of the material. The reader and the lecture slides will allow you for self-study and give you general support to rehearse the theoretical materials. The tutorials, which will be given during the practicum in small groups, will allow you to solve certain problems, to deepen your knowledge, and to ask questions which we can discuss during these tutorials. Finally, a large part of this course is practical. That allows you to do experiments hands-on and also will teach you how to report the results of your experiments. What is bioanalysis? It's the analysis of pharmaceuticals and their metabolites, as well as biomarkers and other endogenous compounds at low concentrations in complex biological samples. To do so, it is necessary to use advanced analytical techniques with high sensitivity and selectivity, as often you have to discriminate your molecule from millions of other, often very similar matrix-derived molecules. You need to optimize the methodology based on a profound knowledge of the physicochemical properties of your analyte, as well as a good understanding of the possibly interfering matrix components. The field of bioanalysis has some history. It started around 1930 with the interest of scientists to understand the toxic effects of compounds. So-called toxicology field required that these molecules be identified and quantified and their concentration related to the toxic effect. Further to this, people were interested in understanding the distribution of pharmaceuticals in an organism, so-called pharmacokinetics, which concentrations are reached and how fast are these molecules eliminated. Unfortunately, the methodology at the disposal was very limited. It was mainly linked to spectrophotometry, meaning the measurement of light absorption of molecules. You can imagine that if you measure in a complex biological sample like blood, that light absorption of a molecule is a not very specific measure to identify and to quantify a molecule. That's why this methodology was limited to high abundance molecules with a very characteristic light absorption spectrum. To move further and to be more specific, separation methods were needed. The first very widely used separation method was chromatography. First methods were based on a stationary phase simply with paper, where molecules migrated in a liquid mobile phase. In parallel, gas chromatography was invented, which was much more powerful, but was unfortunately limited to molecules that could be volatilized, that means that could be transposed into the gas phase. Also around the 1950s, the first ligand-binding assays were developed. They were based on radioactivity, so-called radioimmunoassays. An antibody would capture a molecule, and a radioactive tracer of the same molecule was added in a competition radioimmunoassay. We will go into more details of immunoassays in a later part of the lectures. A breakthrough was the introduction of high-performance liquid chromatography. As you hear, still based on chromatography but with much higher separation power. This was in the 1960s and 70s, based on small diameter chromatography beads that were packed into columns, and these columns were run under high pressure to achieve very high separation efficiencies. In the 1970s, a very important method was introduced. It's called enzyme-linked immunosorbent assay, so-called ELISA. It's also a ligand-binding assays where antibodies bind drug molecules, but the detection is not done by radioactivity, but rather by an enzymatic reaction generating a colored product or maybe a fluorescent product. Again, you will hear more about this in a later part of this lecture. Last not least, in the 1990s, mass spectrometry made its entry into instrumental bioanalysis as a very powerful selective detection method which also provides chemical information about the compounds. Mass spectrometry is a very versatile method because it is not dependent on an enzyme reaction or radioactivity, but it is only measuring mass to charge ratio. You will hear more about this in a later part of this lecture. Finally, two powerful methods, HPLC and mass spectrometry, have been combined to so-called liquid chromatography mass spectrometry combination, which is to date the most powerful combination in instrumental bioanalysis. The field of bioanalysis goes far beyond the pharmaceutical analysis. It's used in many areas of applications. Here are just a few examples. Doping analysis, very well known to most of us, relies very much on high-performance bioanalytical methods to detect drugs of abuse or substances of abuse that may have a performance-enhancing effect. Clinical and forensic toxicology rely heavily on instrumental bioanalysis to detect molecules, to measure biomarkers, and to relate them to toxic effects or to the development of a disease. New pharmaceuticals during their development rely heavily on pharmacokinetic and metabolism studies. These molecules, when they are first used in man, need to be analyzed in terms of their concentrations that are reached, their tissue distribution, their elimination, and especially also their metabolism in view of potentially occurring active or even toxic metabolites that need to be characterized and that form an important part of the regulatory dossier that will go to the FDA or to EMA for approval. Biomarkers are molecules that are used and analyzed in circulation to diagnose or prognosticate disease or to look at the effect of therapeutics. Biomarker discovery is a very difficult field, which we also work on in my group, which means that we want to find molecules that can, at an early stage, predict the development of a disease and hopefully allow preventive measures to be taken in time. Therapeutic drug monitoring is used extensively in university hospitals or in larger hospitals in general. It is related to drugs that have a very narrow therapeutic window, such as immunosuppressants or oncology drugs. These need to be measured accurately and precisely so as not to overdose or underdose the patient. More and more pharmaceuticals are going off patent and are produced by generic manufacturers. However, it is very important to show that these generic pharmaceuticals are equivalent to the original drugs. These so-called bioequivalence studies depend heavily on instrumental bioanalysis. You need to show that the pharmacokinetics and metabolism are the same. You also need to show that tissue distribution is equivalent and, most importantly, related that the biological therapeutic effect is equivalent. Finally, another field unrelated to what we will be discussing here is environmental and occupational safety analyses. That means drugs in the environment, molecules in the environment, and also molecules on the working place. We all have heard about the distribution of antibiotics in the environment, for example, which links drug analysis to environmental analysis. All of these fields require selective, high sensitivity and specific bioanalytical methods, which you will hear about during this course. Like every field, also the field of bioanalysis requires the knowledge of some terms and definitions. First of all, we will speak about qualitative bioanalysis. This means essentially the identification of substances. That means to really be sure that we are measuring the correct molecule. Once we know that, we can move on to quantitative bioanalysis, where we would like to measure the concentration or amount of a given substance in a complex biological material. This complex biological material is often called a matrix. This is the material in which a substance must be determined. A matrix may be blood serum, blood plasma, urine, or tissue sample. In order to develop a quantitative bioanalytical method, it is important to have a blank matrix. That is, the same biological material but without the analyte. This is not always easy to find because some molecules are endogenous and a blank matrix is not available, or molecules are so widely distributed and used that it is impossible to find a blank matrix. In that case, we will resort to other approaches like the standard addition method you will hear about. A calibrator is a known concentration of the analyte of interest in the blank matrix. That means you know what the concentration is, and now you're asking the question, does my analytical method accurately determine this concentration? In order to do so, you need to make a relation between the measured analytical signal, for example light absorption, to the analyte concentration. This is done in a so-called calibration line, which ideally should be linear. In order to account for variations in the analytical procedure, which are somewhat inevitable when you prepare samples, you need internal standards. An internal standard is another substance that resembles the analyte very closely. For mass spectrometry, these are often isotopically labeled standards, and this internal standard then corrects for method variability, for example, to losses during filtration steps or precipitation steps. Here you see a typical calibration line. This calibration line relates the concentration of a molecule in microgram per milliliter to the height ratio of signals. We talk about a height ratio or a ratio in general because the signal of the analyte is divided by the signal of the internal standard, which corrects for variations in the analytical procedure. As you can see, this calibration line is nicely linear, and it is also fairly repeatable as each analysis has been done 4 times, indicated by the 4 dots. You can also see that the variability of the method becomes worse as the concentration increases, while it is fairly repeatable at the lower concentrations. Developing a bioanalytical method starts with the sample preparation. You take a sample, for example a blood sample, and this sample must be prepared to be suitable for the subsequent analytical method. The second step is mostly a separation step, where you try to enrich and separate your molecule of interest from the so-called matrix components. Third is the detection of your molecule. This can be generic detections like light absorption, but also highly specific detection means like mass spectrometry or maybe a ligand binding assay. Finally, the data that you obtained will have to be analyzed and translated into a form so that, for example, a clinician can draw conclusions from those data. Sample preparation is the first step in setting up a bioanalytical method. Often the concentration of your analyte needs to be adjusted to fit the sensitivity of your analytical equipment. In few cases, dilution is necessary if you want to measure high abundance compounds that may saturate your equipment or detector. In most cases, enrichment is needed because we're interested in low abundance, low concentration molecules in complex biological samples. Enrichment invokes also the removal of interfering compounds as well as the capture of the molecule of interest. We will get into more details later on. Phase transfer is an important principle to remember. The transfer of molecules between 2 liquid phases, as in liquid-liquid extraction, between a solid phase and a liquid phase, as in solid phase extraction or chromatography. Many biological molecules, but also synthetic drugs, need to be stabilized to be analyzed. Remember that often samples are taken at different places than where the analysis will take place, so sometimes samples need to be shipped. Freeze drying is an important methodology to stabilize a biological sample by removing water. The dried blood spot technology, which you will also hear about briefly, is another way of stabilizing biological samples. In this case, a small drop of blood of a few microliters is spotted on a kind of filter paper, and thanks to the immobilization, samples can be shipped, often at room temperature, to the analytical laboratory. Finally, adjustment of analyte properties is very important, for example, for the analysis by gas chromatography. For example, sugars, who are inherently non-volatile molecules, can be rendered volatile by derivatization for gas chromatography. So there is much to say about sample preparation. I will go into details about it in a minute. This is an example from our own work illustrating how important it is to standardize and stabilize samples. In this research project we were looking into the effect of the coagulation time to prepare blood serum on certain peptides, the so-called fibrinopeptides, which participate in the coagulation cascade. As you can see, the concentration measured here by mass spectrometry of given fibrinopeptides changes significantly with time and only stabilizes after about 8 hours of coagulation. However, most studies published in the literature work with about 30 minutes to 2 hours of coagulation times, and if this is not really standardized, you can easily imagine that the concentration of these fibrinopeptides will be very variable and that conclusions about their concentrations in the biological material cannot really be drawn. I mentioned already two ways of stabilizing a biological sample. An important one is freeze-drying, especially in larger facilities. In this case, a sample is frozen, here at -5 degrees, and then the water is sublimated into a dry ice bath under vacuum. So the water is transferred from the solid state, the ice state, directly to the gas phase without ever thawing the sample. This often results in a nicely fluffy powder that can be easily redissolved in water or a buffer for subsequent analysis. Another way of stabilization is the so-called dried blood spot collection. You can see how this is done on the YouTube film given below on this slide. Essentially, it is collecting blood on a little piece of filter paper. As indicated on this slide, contamination by your own hands or the external environment must be avoided. Many devices are on the market. They're very simple. This is a so-called finger prick device, where a little needle comes out to poke a little hole in the finger and a few microliters of blood come out. This blood is then spotted on defined areas, so-called dried blood spots. These are dried and then can be shipped in an envelope to the analytical laboratory. The collection of dried blood spots is very popular in metabolic disease of children and also therapeutic drug monitoring, especially in immunosuppressants. Immunosuppressants have to be taken over a long period of time and have to be monitored very carefully. It would be very tedious for a patient to come to the hospital every day for a blood analysis. By using dried blood spot collection, the sample can be collected at home, dried, put in an envelope and simply sent to the hospital analytical lab for analysis. Moving on from sample preparation to separation. Now you've prepared your sample, but it is still a very complex mixture of many different molecules. Some molecules are very similar to each other, for example proteins. They are all built out of 20 amino acids in different sequence orders. So these molecules need to be separated from each other in order to analyze them selectively, as they all give a similar response. Separation has the goal to increase the depth of analysis, especially in the biomarker discovery world, but also in clinical chemistry analysis, where often nanogram per milliliter or even picogram per milliliter sensitivities need to be reached. These sensitivities can only be reached after thorough separation of the molecules and high sensitivity detection. Separation will also improve selectivity, as molecules with similar properties will be separated from each other and thus give different signals in the analysis. For example, different peaks in a chromatogram. Finally, separation is often needed to make samples compatible with mass spectrometry analysis. As you will see, mass spectrometry requires that molecules be transported into the gas phase, into a high vacuum system where the mass-to-charge ratio can be determined. This is not possible when there are too many molecules together ionizing at the same time and competing with each other for transport into the gas phase. This is generally called ionization suppression. An example how important separation is to determine a low concentration of a given molecule, in this case the peptide Salmon Calcitonin in human plasma, is given here. This is a chromatogram where you see on the x-axis the retention time and on the y-axis the ion count, that means the number of ions entering the mass spectrometry detector. Despite a highly selective detector and a very good separation, you can see that human plasma contains many peptides that also give signals in this analysis. Were it not for the separation, the molecule at 2.87 minutes, which corresponds to Salmon calcitonin, could not be discriminated from more abundant molecules eluting at 2.6 or 3.67 minutes. Nota bene, this is a very sensitive analysis. We're measuring calcitonin at the concentration of 50 picogram per milliliter in plasma. This would not be possible without high-performance liquid chromatography separation. Finally, a word about detection. There can be much said about different detection methods, and we'll come back to that in later lectures. However, what is very important generally for good detection methods that they are sensitive, specific for the molecule in question, and in this case this is achieved by a so-called proximity assay. This means a signal is only generated when two molecules are close together. This is a very important detection method for ligand binding assays, as you can imagine, because as the ligand binds to the receptor or the antibody, it generates a signal, while it does not generate a signal when it does not bind. Here on this schematic we see two molecules, a green and a red molecule, binding to the same target. This is double-stranded DNA, but this does not matter really so much for the principle. This binding brings them in close proximity. Thus, when the green molecule is irradiated with green light, it transfers energy to the red molecule, which emits fluorescence at a red wavelength. This is only possible when the molecules are within a couple of tenths of nanometers of each other. As mentioned earlier, bioanalysis can be discriminated into qualitative and quantitative bioanalysis, meaning the identification of substances and their quantification. Any analytical method will result in raw data, and these data need to be analyzed and often compared with statistical methods. So here your knowledge from the Biostatistics course is very important. In the case of biomarkers, we often want to discriminate a diseased group of patients from healthy controls in order to develop better diagnostic and prognostic methods. As you can see here in unpublished work from our group, there's an example between patients and control groups. No matter the analytical and statistical method, you can see that the NP, that means the controls in this case, and the patient's P cluster into different areas of data space, and this means they can be discriminated by different molecules or patterns of molecules. These molecules are indicated as green little stars, and these molecules are increased in concentrations in the patients. When we look at one of these molecules that we identified, then we can see that patients have a significantly higher concentration in urine in this case than controls. The molecule is almost absent in the controls. Such a molecule is a very good biomarker candidate to discriminate patients from controls, but we would have never found it unless we would have statistically compared the very large data sets obtained through urine analysis by liquid chromatography mass spectrometry. If we then plot these results in a very simple univariate statistical approach, we can see in a box plot that controls have a much lower concentration, given here in arbitrary mass spectrometry units, than the patients, and that the p-value is very low, means that this discrimination is statistically very significant. I hope to have given you short examples and an overview of the various steps of a bioanalytical method: sample preparation, separation, detection, and data analysis. However, now the question arises, how do I choose the correct bioanalytical method and how do I develop such a method? There are a couple of considerations that are important before starting. First of all, what is the goal of the analysis? Is it simply to identify a molecule, or do you also need to quantitate? Look at the physicochemical properties of your analyte. What is the structure? Is it an acid? Is it a base? Is it Zwitter ionic? Is it a large macromolecule or a small synthetic molecule? What is the biological matrix I have to measure in? Is it highly complex like blood, serum or plasma? Or very diluted like urine? Or do I need to work in tissue which I have to first prepare and homogenize? Which concentration range do I need to cover? That means at which concentration is the molecule likely to be biologically active or relevant? That translates into the required sensitivity of your method. It is very different to develop a method that has to measure a microgram per milliliter or a picogram per milliliter. What is the required throughput? This will define the technicalities of your method. Do I need to analyze 1,000 samples per day or maybe 100 samples in a year? For 100 samples in a year you can use a method that is very much manual, while with 1,000 samples a day you need an automated method. In the current environment you always have to think about the cost. How much can I charge for one analysis? How much may it cost, for example, in view of the health benefits and what the health insurance may be willing to reimburse? And finally, and not to be forgotten, what is the available instrumentation in the lab and do I have trained personnel? Actually, not every lab is equipped with a mass spectrometer, so it makes no sense to develop a mass spectrometric method if you do not have the correct instrumentation. More importantly, for complex methods such as liquid chromatography, mass spectrometry, it is very important to have trained personnel. Trained personnel is not available very easily, and it takes time to acquire this knowledge to perform these methods. Trained personnel to date is often the bottleneck of implementing modern bioanalytical methods in routine laboratories. Let's move on to some concrete examples of biological matrices. We've already talked about blood, plasma and serum quite extensively. They are probably the most widely used biological matrices. Urine is often used. It's easily obtainable, but it's very variable in its biological composition and needs to be collected very carefully, in a sense that it is not contaminated and that it is also not too much affected, for example, by the nutritional status of the person. Cerebrospinal fluid is collected in special occasions, especially in the context of neurological disorders. However, it is a quite invasive procedure and it cannot be done on a routine basis. The same goes for bronchoalveolar lavage, which is collected from the lung during bronchoscopy. This is also a fairly invasive procedure requiring anesthesia and is thus not useful for routine diagnostics. Hair, on the contrary, is easily to obtain. Hair can accumulate, for example, substances of abuse over many months and can be used for retrospective analysis of drug or substance abuse. Tissue can only be obtained by biopsies and is thus not widely used for diagnostic procedures unless a biopsy can be taken. Biopsies are often taken during operation, especially in oncology, where tissue is available and widely used for diagnostic procedures. But let's focus first on one of the most widely used biological matrices that you will encounter as well: blood plasma. Plasma, after centrifugation of blood, you will find a cell pellet and an upper layer which is kind of murky. That's called plasma. However, in order to obtain plasma, you need to anticoagulate blood. That means you need to prevent blood coagulation, as otherwise you would obtain serum. Blood coagulation can be prevented in different ways, and these different ways may have an effect on the composition of plasma. A widely used way is to include EDTA, a chelating molecule that chelates calcium. As calcium is chelated away from the blood coagulation cannot proceed as all the coagulation factors require calcium. Other ways to anticoagulate is heparin. Heparin is an inhibitor of thrombin, and thrombin is the key protease in blood coagulation. So as you inhibit thrombin through heparin, blood cannot coagulate and you can obtain plasma. Now it is important to understand the composition of your matrix, in this case plasma, as much as it is important to understand the physical-chemical properties of your analyte. Plasma is a very protein-rich biological sample. You can see here there are 60 to 80 grams per liter of protein. It also contains quite a high ionic strength, as the milliequivalents per liter of cations is about 150 millimolar. Total lipids can vary a lot in plasma, but is usually between 0.2 and 0.8%, which is mainly phospholipids and triglycerides. Cholesterol forms a high percentage of the lipids in plasma, the normal concentration being below 5.2 millimolar. As you know, cholesterol does not occur free, but is bound to high or low density lipoproteins. All these biological components of plasma may affect your analytical method and have to be taken into account during sample preparation, separation, and detection. Protein binding is a very important property of low molecular weight pharmaceuticals. Here we have 3 examples of basic drugs which have very different levels of protein binding. While 2 of them bind to more than 90% to proteins, mainly albumin in plasma, one, amphetamine, does not bind at all or to a very low extent. This affects the physicochemical properties of these molecules significantly, as they are transformed from low molecular weight drugs to high molecular weight drug protein adducts. Why is it that different molecules bind differently to proteins? Honestly speaking, there is very little known about the molecular properties of molecules that determine protein binding. So let's look at a few structural features. Acetazolamide is a complex heterocyclic molecule with an amide bond and a sulfonamide bond, providing many possibilities for hydrogen bonding. This could be one reason why it binds to proteins so strongly, because proteins also contain many hydrogen bond donors and acceptors. Amphetamine is bound very weakly to proteins. It is actually a very simple molecule, only containing one amino group and an aromatic ring, and in the rest a fairly hydrophobic backbone. This may mean that hydrogen bonding is not very likely with amphetamine, and protein binding is consequently rather weak. Amitriptyline again has high protein binding and is also a basic molecule. Amitriptyline also has one tertiary amine, but a much larger conjugated aromatic system. This may mean that it can interact better with aromatic amino acids, which leads to more protein binding. As you can see already, this is all rather unclear, and protein binding cannot be easily predicted. That means it should be and it must be measured by bioanalytical methods. Here is a setup how to measure drug protein binding. The method is called ultrafiltration. The center part of the ultrafiltration device is a membrane. This membrane, depicted here, is fixed between an upper reservoir, the sample reservoir, and a filtration cup. All molecules that should enter the filtration cup have to pass through the membrane. This membrane has defined pore sizes so that big molecules cannot pass while small molecules can. Imagine our blood sample with a drug molecule in it that partially binds to proteins. Proteins are large molecules that cannot pass through the membrane, such as albumin, while the free drug molecule is small and can pass through the membrane. Putting such a device in a centrifuge to apply some force, so to force the liquid through the membrane, makes it easy to measure protein binding by determining the drug concentration in the upper bound fraction, the protein-rich fraction, and in the filtrate, which is protein-free and contains the free drug. Another method to measure protein binding is dialysis. It has a similar principle. It constitutes a membrane with pores of defined size that does not let pass proteins, and an external volume, for example a beaker. Finally, if you measure the bound drug concentration inside the dialysis tube and compare it with the concentration outside, you can get a ratio between free and protein-bound drug. Let's end this overview with a final aspect of drug bioanalysis, drug metabolism. As you've already heard, drugs are metabolized in phase 1 and phase 2 reactions to excrete them from the organism. Some of these drug metabolites may still be active. It is even so that some drugs are inactive, and only once they are metabolized they become active. A known phase 2 reaction is the conjugation of drugs. In this case you see morphine, a widely used painkiller, conjugated to glucuronic acid, a carbohydrate, to make it more hydrophilic and thus more easily excretable through the kidneys. Morphine 3-O-glucuronide is active and can accumulate in the circulation, so it must be monitored carefully. There are enzymes that can deconjugate morphine from the glucuronic acid, make it possible to measure the drug metabolite 3-O-glucuronide and the free morphine separately, provided you have a bioanalytical method that can discriminate the two, for example a separation by liquid chromatography. With this, I hope to have given you a good overview over the various aspects of bioanalysis, from sample preparation via separation and detection to data analysis. Instrumental bioanalysis depends on technology developments and understanding the analytical technology, as well as understanding the applications and the goal of the analysis, be it clinical, chemistry analysis and diagnosis, therapeutic drug monitoring, or the development of new pharmaceuticals. We will now move on to more specific aspects of instrumental bioanalysis.